Write the difference between nucleoside and nucleotide. Nucleoside, it is the combination of nitrogenous base and a pentose sugar. Example is adenosine, guanosine, thymidine, cytidine, uridine. And upon phosphorylation, nucleoside can be converted into nucleotide. And a nucleotide is can be defined as it is a combination of nitrogen base plus pentose sugar and as well as a phosphate moiety. The difference between nucleoside and nucleotide is only the phosphate moiety. So phosphate moiety is the difference between the nucleoside and nucleotide. Nucleotides are building blocks of nucleic acid, that is DNA and RNA structure. So these are structural components of coenzymes of B complex vitamins. So certain nucleotides can be considered as coenzymes such as FAD, NAD, nicotinamide adenine dinucleotide ion. So NAD plus. So like that, they can be acts as a coenzymes also. So adenosine monophosphate, guanosine monophosphate. What are purines and pyrimidines? These are two kind of nitrogen containing bases are called as purines and pyrimidines. Purines, which are consists of 6-membered nucleus and 5-membered nucleus, which are containing the nitrogen, and the rings are fused together. Examples are adenine, guanine. So these are purines and pyrimidines. Hypoxanthine, xanthine can also comes under purines category. So the structures of adenine and guanine, if you observe that, so 6-membered cyclic structure having nitrogens can be fused with that of a 5-membered cyclic structure having 2 nitrogen atoms. So pyrimidine nucleus fused with that of imidazole nucleus in the structure of adenine, and in the guanine also the same thing can be observed, but the positions can be differ. In that sixth position is having of amine group in the case of adenine. Here in the case of guanine, so ketone group is present. At second position amine group is present. Pyrimidines, so these are pyrimidines, they have only a 6-membered nitrogen containing ring. Examples are uracil, thymine and cytosine. Uracil, which is present in the RNA only. So thymine, cytosine can be absorbed in DNA and RNA. Here the adenine and guanine can be absorbed in the DNA as well as RNA also. Examples for pyrimidines are uracil, thymine, cytosine. The structures are as follows. So thymine, cytosine and uracil having of only one heterocyclic ring system having nitrogens. Write a short note on hyperuricemia and gout. Hyperuricemia, that is elevation of serum uric acid concentrations more than 7 mg per dl, is known as hyperuricemia. So uric acid is the end product of purine metabolism. So what is mean by gout? So gout is a metabolic disorder which is associated with the overproduction of uric acid. So overproduction of uric acid may causes gout. So this may causes gouty arthritis, that is inflammation of joints. Draw the diagram or structure of DNA and RNA. So in the case of RNA, you can draw the tRNA structure. DNA is having double strand, so and it is appears like a ladder like structure. So double helical structure of DNA, here you can see that so each helix having of 34 angstroms length, and 10 base pairs are present. So each base pair can be having of space 3.4 angstroms. So the width of the DNA is 2 nanometers. So the DNA one strands can start from 5 prime, another strand starts from 3 prime. Then coming to the structure of tRNA. So it is having of various arms. So the acceptor arm, D arm, T tau C arm, variable arm, at the same time anti-codon arm. So like that it is having of various arms, and each arm having a complementary base pairs, and the number of complementary base pairs can differ from one arm to another. Mention a short note on Chargaff's rule. The content of adenine equals to that of thymine and guanine equals to that of cytosine is known as Chargaff's rules. That is, number of adenine moieties present in the structure of DNA can be equals to the number of thymine, and number of guanine molecules can be equals to that of number of cytosine. Define DNA replication. So DNA replication is the process in which DNA copies itself to produce identical daughter DNA molecules. It is essential for the survival of the species. So the production of DNA from DNA is known as DNA replication. Define transcription. So transcription is a process in which RNA can be synthesized from DNA. So the genetic information stored in DNA is expressed through the RNA. So production of RNA from DNA is known as transcription. Define translation. The biosynthesis of protein or polypeptide in living cell is known as translation. So in the translation process, mRNA can produce the protein. The genetic information which is stored in DNA is passes to the RNA and ultimately expressed in the language of proteins. Give examples for inhibitors of protein synthesis. Antibiotics which inhibit the protein biosynthesis in the bacteria and prevent the growth. Examples are streptomycin, tetracycline, polymyxin, chloramphenicol, erythromycin, etc. Write a short note on genetic code. The three nucleotide base sequences in a mRNA that act as code words for the amino acid in protein biosynthesis is known as genetic code, or simply codons. So the three nucleotides which can simply called as a triplet. So genetic code is a dictionary of nucleotide bases, that is adenine, guanine, cytosine and uracil. So that determines the sequence of amino acids. Codons are two types. One is initiating codons, second one is termination codons. AUG, GUG are examples for initiating codons. UAA, UAG and UGA are the termination codons. The termination codons which do not have matching amino acids. That's why the protein synthesis stops whenever the UAA, UAG, UGA comes into the sequence of mRNA. Mention various types of RNAs and its functions. So we have various types of RNAs, that is mRNA, hnRNA, tRNA and rRNA. So mRNA means messenger RNA. Transfer genetic information from gene to ribosomes to synthesize proteins. hnRNA is also known as heterogeneous nuclear RNA. It serves as precursor for mRNA and other RNAs. Next one is tRNA, also known as transfer RNA. It transfers amino acid to mRNA for protein biosynthesis. rRNA, also known as ribosomal RNA. It provides structural framework for ribosomes.